Okay. Prophase is the first phase in mitosis. It's when the chromosomes start to coil up. And then metaphase is the second phase where the chromosomes meet at the equator. And then anaphase is the third phase. And that's when the chromosomes start to move to opposite poles. Telophase, two nuclei form, and they start to split, and then in cytokinesis is when they actually split and become two new cells. Then it goes through the G phase of interphase, and then in the S phase, the DNA go through semi-conservative replication, where it keeps one strand and gets a new one. And then
there's a change in gene mutation. A mutated gene is called an oncogene. And cancer results when cell undergoes uncontrolled mitosis. The cells divide out of control and result in a tumor. Any mutation can be a mutagen. Any other factor that causes cancer is carcinogenic.
Okay, so this time we have a DNA, but it's coded for hemoglobin with sickle cell anemia.
tragic.
So this is HBS, and as you can see, the messenger RNA will come take the code from the codons, but instead of the glutamine, there is the valine at the end. So then the messenger RNA will leave with the sickle cell anemia trait and go to the transfer RNA, where this transfer RNA is not correct, and it will code for the wrong amino acid. So then these will again go up, grab their amino acids, and as the and so then the messenger RNA will go through the ribosome. So then the messenger RNA will go through, and the transfer RNA will then bring in the amino acids as they go. And as they all go through, the amino acids will form peptide bonds to form the hemoglobin with the sickle cell, which then looks like this guy over here with the wrong amino acid right here. Instead of glutamine, it is a valine. And it doesn't sit right, so it falls off.
As I go back to the...
Alright, so here we've got the cell cycle, and within prophase, metaphase, anaphase, and telophase, mitosis occurs. Then
there's cytosis, where it splits the cell into one old cell and the new cell.
Then throughout the rest of interphase, mRNA is created from DNA. Okay, so then the DNA is unzipped, which creates the mRNA, and then the mRNA leaves the nucleus and heads over to the tRNA and creates the tRNA actually, which then goes to the ribosome, the mRNA comes over here, and then once the tRNA is created, then the tRNA will correspond with its correct amino acid, and after that point, the tRNA and mRNA will both go to the ribosome, which is right over here.
Got cancer.
Just cut it off. I don't even know.
I can do this.
Alright.
So it's not there.
Alright, so after the mRNA and the tRNA are going through the ribosome, they create they will then create the primary structure, which will be created from peptide bonds
Of amino acids.
Yep. And then those peptide bonds are gonna go and create a tertiary structure, secondary, whatever. You know what I mean?
Where it bends into its state of hemoglobin with the alpha helix, kind of, trying.
Imagine.
Now.